Welcome back to our third and final installment of this section on the regulation of the cell cycle. So if cells proceed from one phase of the cell cycle to the next without completing the previous step, then the resulting daughter cells could be abnormal. For example, aneuploidy, which is an incorrect number of chromosomes, could result if DNA replication isn't completed correctly. So cells use a series of checkpoint mechanisms to ensure that each phase of the cell cycle is completed properly before it goes on to the next stage of the cell cycle. So there are several of these checkpoints. We'll look at maybe 5 different types of checkpoint. First one is to look at the G1S checkpoint. And the key players here are the CDK cyclin families that accumulate during G1, and a protein called RB and a protein called E2F. So in cells which are receiving the appropriate signals from growth factors and nutrients in the environment, the RAS pathway is activated, and that turns on the CDK cyclin pathway. And so we have an accumulation of active CDK cyclins when there are adequate growth factors and nutrients in the environment. So what that means is that normally CDK and cyclins can phosphorylate this protein called RB. Now, before RB gets phosphorylated, it's tightly bound to this protein called E2F. And E2F is a transcription factor which would normally be driving the expression of genes required for cells to go into and through S phase. So when RB is bound to E2F, those genes cannot be transcribed. So along come the CDKs and cyclins, they phosphorylate RB, RB releases E2F, E2F stays bound to the DNA, and you get transcription, subsequent translation, and you get the enzymes and other proteins required for S phase. And so this checkpoint works if there are no growth factors. So if we don't have no growth, if we have no growth factors or inadequate nutrients, then the CDKs and the cyclins don't get turned on, which means that RB stays unphosphorylated and bound to E2F, and therefore there is no expression of the genes required for S phase. So that gets us into S phase. And so then there's a checkpoint which regulates whether DNA replication or regulates DNA replication initiation. So it's crucial that DNA replication occurs only once, so that DNA molecules are replicated once and only once. And that is ensured through a process called licensing. And so licensing is under the control of these proteins called MCMs. And so MCM proteins bind to sequences on DNA called the origins of DNA replication. That's why DNA replication initiates. And so the binding of MCMs to those origin sequences is dependent on a protein called the ORC, the origin recognition complex, and then some proteins which load MCM onto the DNA. And then once DNA initiates, the MCMs and the other proteins on the initiation complex are displaced from the origin. And then re-replication or re-initiation of replication is prevented by stopping the MCMs coming back onto the DNA. So the MCMs can bind onto the DNA, and then you initiate DNA replication, which displaces them, and then you just stop them coming back on, and that allows DNA replication to initiate once and only once. So key player, as you might expect here, is the CDK family. And so the CDKs block relicensing by inhibiting the function of proteins such as ORC and helicase. So once the CDKs become active, they block the reloading of MCM onto the origin of replication. There's another protein called geminin, and so geminin works with CDKs, and gemin is made during S phase, and that also blocks the loading of MCMs onto the origins. And so once geminin is accumulated, it's blocking the reinitiation of DNA replication. And then when you hit M phase and the APC gets activated during anaphase, that breaks down the geminin, and that means geminin is no longer present, and therefore it can't block the loading of the MCMs. So that's the regulation of DNA replication initiation. Then there's a checkpoint at the G to M transition, which basically asks the question, is DNA replication complete? That makes sure that every single nucleotide has been replicated before the cells go into G2. You don't want to be going into G2 with an incompletely replicated genome, because that will lead to aneuploidy. And so what happens here is that the final dephosphorylation step in activating the CDKs doesn't happen. So if you remember back to our discussion of that. Cyclin levels go up as you're about to go into mitosis. The cyclin binds to the CDK. You have 2 inactivating phosphorylations, an activating phosphorylation, and then you take off the 2 inactivating phosphorylations. And so this checkpoint stops these inactivating phosphorylations from being removed. And so you end up with a kinase which remains in an inactive form as the cells are blocked from going into mitosis. The fourth kind of checkpoint we can take a look at is called the Spindle Assembly checkpoint, also known as the mitotic checkpoint. And this prevents anaphase from occurring before all of the chromosomes are attached to the spindle. So kinetochores that remain unattached to microtubules produce kind of a wait or a pause signal that inhibits the anaphase promoting complex. And the main players here are, of course, the anaphase promoting complex down here, a protein called CDC20, and then the MAD and the BUB proteins. So MAD and BUB are proteins which accumulate at the kinetochores of unattached chromosomes. So this kinetochore here is unattached, so MAD and BUB proteins accumulate. And then they become part of a complex involving, including this protein here, CDC20. So they sequester CDC20, and what CDC20 does is it activates the anaphase promoting complex. So if CDC20 is bound by the MAD and the BUB proteins, then the APC cannot be activated, so you cannot initiate the activation of APC. That means you can't do the degradation of that cohesin protein, which is involved in separating or allowing the separation of the sisters. So once the microtubules attach to kinetochores and become kinetochore microtubules, this association between the MAD and the BUB complex is lost. So the MAD and the BUB proteins dissociate from the kinetochore, and that frees CDC20 to bind to the APC, and that switches the checkpoint off. So as long as MAD and BUB are stuck on the kinetochores, you will keep cells stuck at basically metaphase. And then as soon as everyone's attached and there's tension in the spindle, you'll get release of CDC20 and activation of the anaphase promoting complex. Last checkpoint to look at here are a series of checkpoints, actually, which monitor for DNA damage. And so there are multiple DNA damage checkpoints that monitor the integrity of the genome. Then they halt the cell cycle at various checkpoints. There's a late G1 checkpoint down here. There's an S-phase checkpoint. And then there's a late G2 checkpoint. And each of these checkpoints works through different CDKs. So this entry into M-phase checkpoint is controlled by CDK1, and then this S-phase entry checkpoint over here is controlled by CDK2. Possibly the most important protein here is this protein called P53, also known as the guardian of the genome, lost in up to 50% of human cancers, so that's a super important protein. So, for example, double-stranded breaks in DNA, which are highly toxic, activate this protein kinase here called ATM. And then ATM activates this protein called CHEK2. So CHEK2 is called a checkpoint kinase, and CHEK2 can phosphorylate P53, and that activates P53, which is a transcription factor, and stimulates the production of a protein called P21. And then P21 inhibits CDK2, which is one of the CDKs controlling entry into S-phase. So, what CDK normally does is phosphorylates the retinoblastoma protein. And if you remember back a video or two, retinoblastoma binds to the E2F protein, and when RB is bound to E2F, E2F cannot initiate transcription of the genes involved in going into S-phase. So if you inactivate CDK2, you cannot phosphorylate RB, and therefore you cannot activate E2F. So by turning off CDK2, transition into S-phase from G1 is blocked. So you get this cell cycle arrest in late G1, and that allows the cell to repair the double-stranded break damage, and if it can't do that, it can trigger apoptosis or programmed cell death. Other types of DNA damage activate the ATR kinase, and that turns on this checkpoint kinase, CHEK1, and CHEK1 can turn off CDC25. So if you again think back to what's happening with the CDK as we go into mitosis, cyclin levels go up, cyclin binds to CDK, 2 inactivating phosphorylations, an activating phosphorylation, and then normally that CDC25 protein takes away the 2 inactivating phosphorylations and you're left with an active CDK. So if you inactivate this phosphatase here, that means that CDK1 cannot become activated, and you don't get entry into S phase, into M phase. Sorry, so you get cell cycle arrest. This happens in S phase, but we're talking about in G2M here, and so that can allow for DNA repair. Or again, if we can't fix the DNA damage, you end up getting apoptosis. That's it for regulating the cell cycle. The last video we'll look at will be a process called apoptosis, or programmed cell death. I'll see you for that soon.